Okay, so today I'm going to talk about Hardy-Weinberg principle for the Khan Academy Indian Talent Search. Well, this is a topic that seems a bit abstract to a lot of students, and they don't understand its use in real life, so hopefully I'll be able to help. Okay, so this principle was given by the British mathematician G.H. Hardy and German physician Wilhelm Weinberg. So the principle basically says that the allele frequencies in a population are stable and constant from generation to generation, and the gene pool remains constant. This is applicable when there is genetic equilibrium. That is, when the population under study is not evolving. We'll first try to understand this whole statement using an example. Okay, we have a hypothetical example where we have, let's say, 100 beetles in a park, and we are studying the gene for color of the beetles. So we have two alleles, big R and small r. If the genotype is big R, it means the beetle is red. Small r means white, and big R, small r means pink. And this is obviously an example of incomplete dominance, where one allele is not able to express completely over the other allele. That's why we have big R, small r as pink and not as red, which would have been applicable if we were talking about complete dominance. Okay, so in this example, we let's say have 40 red beetles, 10 white beetles, and 50 pink beetles. Okay, so beetles are diploid organisms, that is, they have two sets of DNA. That means if we have 100 beetles, so that means that we are going to have 200 copies of the gene that we are studying for the color. And out of these 200 copies, there are 130 copies of our allele. 130. How did we come at to that conclusion? Okay, so I have this one written here. We have 40 red beetles. So if they are red, that means they are big R. So from this we get 40 times 2, we get 80. If we are white, we have smaller, so from this we get 0. And we have 50 pink ones, that means big R, small r. So we are going to have only 50 alleles, which are R. So we get 130 copies of R allele. And if we talk about the frequency of the R allele, we get 130 over the total number of copies, that is 200, gives us 0.65, or we have 65%. Now we know that the sum of allele frequency is 1. That means if we have 65% of R alleles, then obviously, since the other option is only small r allele, so that means that for small r we're going to have a frequency for small r is going to be 35%. Let me write big R here and small r. Okay, now sum of allele frequency is 1, so that's how we got 35% of smaller alleles. Now, what are the possible genotypes that we talked about? It is big R, smaller, and big R, smaller. So let's try to find the probability of getting those different genotypes. Okay, so let's talk about probability of getting genotype big R. So the genotype probability will be R square, that is R times R, because for the two places we're going to have the same big R allele. So we know that frequency for the big R allele is 65%, or 0.65, and again times 0.65, so we get 0.4225, or 42.25%. So 42.25% of next generation beetles will have bigger genotype. Next is probability for genotype smaller. That's going to be obviously like the previous one, small r square, and frequency is 0.35, so you get 0.1225 or 12.25%. So 12.25% of next generation beetles will have small r genotype. Now the next one, that is RR heterozygote, this can occur in two ways. See, one way is if we get sperm which has big R and egg has small r allele, or the other combination which is possible is that sperm is small r and egg is big R allele. So we get two cases. So for the first one we can write R times R, and for the second one we have R times R. We add the two probabilities to get the final one. Twice of R big R times small r gives me 2 times 0.65 times 0.35. Gives me 0. and 45.5%. Okay, so we have probabilities for all the genotypes, and since we have stated earlier that sum of allele frequencies is 1, and that means genotype frequencies must also add up in the next generation. We are not losing alleles in the next generation, and there's no change in the alleles, so obviously the genotype frequencies must add up to get 1 or 100, I mean if you're talking about the percentage. So if we add all of them, what we get is, we had 42.25%. For the red one we had 12.25. For the pink one, sorry, the white ones, and for the heterozygote we have 45.5%. It all adds up to 100%, so our calculations have been right. So there is a simple formula that you can use to find out whether the population that you're studying is in Hardy-Weinberg equilibrium or not. So what we just did, the example, we know that the genotype frequencies all add up to 1. So we've written all the genotype frequencies here. Big R squared gives us the number of red beetles, 2 big R small r gives us the pink beetles, and small r squared gives us the white beetles. And we know from mathematics that a plus b whole squared gives us a squared plus b squared plus 2ab. So we can turn this equation into this equation, big R plus small r whole square equals 1. This is a form of equation that you might, with these two variables, p and q representing different kinds of alleles, you add them up and you get 1. So that is something that you can use. Okay, so now we talk about the uses of Hardy-Weinberg equilibrium. One is where you have assumed that you have Hardy-Weinberg equilibrium, and using that, then you find the genotype frequencies as we just did in the example. The second is that you know you have the genotype frequencies, and then using those frequencies you try to see whether you have Hardy-Weinberg equilibrium or not. If yes, you have it, then you have a not evolving population. So let's so for the beetles, if we say that they're in Hardy-Weinberg equilibrium, that means that the gene for color is not evolving. The allele frequencies are constant. And if you do not get equilibrium, that simply means that the population is evolving. It also has a medical application, and it helps us to estimate the percentage of people who have a genetic disorder like phenylketonuria or cystic fibrosis in a population. So the conditions, there are certain conditions that need to be there for Hardy-Weinberg equilibrium. So these are the ones. There should be no mutation, there should be no there should be random mating, no natural selection, no genetic drift, no gene flow, and if there is any deviation, if one of them isn't there, then that's going to lead to evolution. Thank you so much for watching, and I hope it helped.